Metabolic engineering is a technique which uses genetic engineering to modify the metabolism of the organism in order to increase the production of certain substances inside the cell in a cost-effective manner. The most common examples are the production of beer, wines, cheese, pharmaceuticals, and other valuable biotechnology products using engineered cell factories. In the past, production of certain metabolites included a chemically induced mutation to genetically modify the organisms. The mutant strain with the desired metabolites was then chosen. However, the metabolic pathway and enzymes involved in the production were unknown, so modifications of the metabolic pathways and enzymes was not possible. During the 1990s, the discovery of genetic engineering and synthetic biology helped to overcome this problem, and so now it was finally possible to analyze each metabolic pathway and enzymes. Synthetic biology and metabolic engineering are two interdisciplinary fields that need one another. Synthetic biology is the fundamental use of synthetic DNA, genetic circuits, or molecular switches, which can be used in the metabolic engineering for controlling the metabolism in cells suitable for the commercial purpose. The production of sustainable ornamental plants is a good example of the integration of these two fields. In an article published in 2012, it has been reported that the worldwide production value of ornamental plants comprises about 50 billion euros, and an estimated global consumer consumption lies between 100 and 150 billion euros. In the same article, it was also reported that consumers prefer short, compact plants as they can tolerate mechanical handling and transportation is also very easier. Furthermore, smaller plants require less space. They are easier to handle with reduced transportation costs. So scientists started to think how to make plants smaller. In many researches, it was found that gibberellic acid plays a major role in the growth of plants. This is why metabolic engineering can be utilized by modulating the biosynthetic pathway of this hormone, which can result in a compact plant. The biosynthesis of gibberellic acid is a very complex pathway, but in this part of the pathway it is quite noticeable that gibberellic acid 20 oxidase plays a major role in the synthesis of various structures of gibberellic acid. And so, researchers has proposed a molecular engineering approach to block the biosynthesis of gibberellic acid by targeting this enzyme. Using synthetic biology, the ornamental crop Blossfriedliana was genetically modified by introducing an alcohol-inducible promoter system that silenced the expression of GA20-OX enzyme. In normal pathway, QBGA20-OX gene that codes for GA20-OX enzymes is responsible for the biosynthesis of gibberellic acid, which then causes the plant to elongate. Using Acurobacterium, an alcohol A-inducible promoter sequence was introduced into the small leaves of this plant. An induction with only 2% alcohol can then downregulate the gene and block the pathway, causing a significant decrease in the height of the transgenic plants by 63 to 90%. In the above example, we have seen that the metabolic engineering was about blocking a metabolic pathway. This is only one of the strategies used for metabolic engineering. Other strategies are overexpressing the gene encoding for a rate-limiting enzyme, heterologous gene expression, enzyme engineering. Now let's look at another example that uses the first two strategies. Metabolic engineering has been carried out in Saccharomyces cerevisiae to produce artemisinin, which is an anti-malarial drug. Artemisinin is used to be extracted from Artemisia annua, which is commonly known as sweet corn wormwood, and is known to be highly effective against the multi-drug resistant Plasmodium species. However, it is short in supply and not affordable by most malaria sufferers. This can be overcome by introducing a whole new metabolic system in yeast so that the yeast can produce the drug in large quantity and in a cost-effective manner. In this simplified metabolic pathway, the initial precursor of artemisinin is farnesyl pyrophosphate, which is already produced by the yeast. In this research, scientists have managed to engineer the metabolic pathway by three steps. First, they engineered the APP biosynthetic pathway inside the yeast in order to increase the APP production and decrease its use by the yeast itself. Second, they introduced an ADH gene taken from the sweet wormwood plant to convert FPP to amorphidine. And last, they introduced a second gene for cytochrome P450 that performs a three-step oxidation of amorphidine to artemisinic acid. For the first step, the upregulation of FPP was done by integrating an additional copy of truncated 3-hydroxyl methylglutarol coenzyme A reductase gene, an ERG20 gene, into the chromosome of the yeast. The usage of FPP by the yeast was inhibited by downregulating the ERG9 gene using a methionine repressible promoter. In the second step, heterologous expression of ADS gene in the yeast was done by extracting the gene from wormwood plant, which then catalyzes the conversion of FPP to amorphidine. Finally, two genes for P450 extracted from the plant was expressed in the yeast, which then metabolically synthesized artemisinic acid from amorphidine via the three oxidation steps. And this is how a whole new metabolic pathway could be engineered into a totally different organism to serve a particular purpose.